Can everybody see that in presentation mode now?
Yep.
Good, okay. All right, well, thank you everybody for being here. I'm Christa Reeves. I'm the Water Quality Program Coordinator with the MUSCONETCONG Watershed Association, and I'm going to discuss water quality monitoring we did from tip to stern, basically, in the watershed from Lake Apacong all the way down to below Bloomsbury for harmful algal blooms. This is the first year that we've done this monitoring, and I'm going to dive right in. All right, so I know a lot of you know this, but I'm just going to go through some of the background. You know, River Watchers is a program we've had at the MWA. It's in its 14th year for citizen science monitoring. We sample 7 sites 4 times a year, every year. Even through COVID, we got a couple of samplings in. I'm very proud of that. And of course, the benefits to citizen science is that, you know, average citizens get to see changes in the watershed, you know, the land and how that affects the river and its health when they wouldn't otherwise be looking that closely at something like that. They can also measure the value of a restoration. There's been several sites like the Hughesville site, you know, where we have an Enviro DIY station that we can monitor that restoration in action. And then also, as many of you know, we have an invasive snail called the New Zealand mud snail. So we're always on the lookout for the New Zealand mud snail and whether it's spread to new sites or not. And then just always getting citizens involved with the river and outside and to become an advocate, really. So the background for this study is that basically Nancy Lawler and I were at a New Jersey water quality monitoring meeting, one cold, I think it was January or November, and they were giving a lecture, you know, series of lectures on harmful algal blooms. And I turned to her and I said, I feel like, you know, we should get into this. You know, like I think this is something we have the capacity to do. It's an emerging water quality contaminant that we're not looking at. And this sounds like a good project for us. So I applied for a Watershed Institute grant and got that. So we were able to purchase 4 handheld meters, the same brand and exact model that the EP uses, so that all that data was comparable. It's called the Turner fluorescence. We're also able to have time to research this and, you know, become knowledgeable ourselves, and then to be able to create a training that volunteers could then turn around and understand and use in the field. With these meters, they measure phycocyanin, so that is the pigment that the algae gives off. It's not a direct measurement of this algae, it is an indirect measurement of the pigment. And then we're also measuring for water temperatures from June to September. A couple other readings, supplemental samples were taken from students at Centenary University to look at some other cofactors like phosphorus, nitrogen, and pH. And we'll talk about that a little bit later. So what is a HAB? What's a harmful algal bloom? So, you know, as climate change keeps marching on, we're seeing more and more of this pop up in the news. It's a certain type of cyanobacteria. It's not actually an algae in a water body that tends to surge in response to certain environmental conditions. Usually it's water temperature, nutrients, mostly phosphorus, sometimes nitrogen. Flow of the water prefers slower moving and even stagnant waters, and sunlight. So not an algae, why are we calling it harmful algal blooms, right? Well, I think it's just an easy acronym for people to use, but let's get used to calling it cyanobacteria so that we're correctly identifying what it is. Now, it's not all bad, even though it's causing us some problems. We wouldn't have an atmosphere if it weren't for cyanobacteria. It's pretty much the most important part of the early formation of our biosphere. And it's been around way longer than us, 2.4 billion years. So what makes this reactive to sunlight is that it's photosynthetic, and those are those photosynthetic pigments inside that is detected by the handheld meter. So where might you find one of these HABs? Well, as anybody who lives up and around Lake Musconetcong or Lake Hopatcong knows, you're going to find it mainly in lakes or reservoirs. A lot of our drinking reservoirs are now becoming subject to blooms of these HABs. Like I said before, it requires slow moving or non-moving, but we have been finding them in areas where the current is more than what I would call a lake, let's put it that way. It's less common, however, in rivers and streams, but possible. That, as shown in this picture, this is actually a tributary to the Musconetcong that has some HABs formed in it. So why are they a problem? Like I said, it's just a natural part, of our atmosphere and our ecosphere. So, but what's the problem? Well, when they get out of hand on a constant basis, like they have been, mainly in the last 10 years or so, they can be harmful. They can cause respiratory irritation, skin rashes if you come into contact with it. If you're out there on a wave runner and it, sprays up on you, you can get some type of contact dermatitis from it. If ingested, you can, especially children or people who are have compromised immune systems. It can cause flu-like symptoms, liver toxicity, certain types of neurological effects. And then, of course, it has the other impacts, such as negatively affecting businesses. The economy, in Lake Hopatcong took a pretty big hit in 2019 when this first became an issue. And then, of course, there's an increased cost to processing that water for drinking, like in the reservoirs, or it can kill or sicken some animals. So like you see that pup up there in the corner swimming in this stuff, we don't want him to be drinking any of this. And if you have animals and there's any kind of bloom in the area, please do not let your dog go into the water. So we're gonna do a quick how to tell a HAB apart from a lookalike. So HABs can appear in several forms, but they all break apart when disturbed. So they're single-celled organisms, but they like to organize themselves a lot of times. So you'll see in the upper left-hand corner that looks like green dots. Some people call it pea soup, or it can look like that streaked paint in the lower right-hand corner, or it can look like streaks, parallel streaks in the lower left-hand corner there. And surface scum. Sometimes it can look like surface scum. It depends on, which genus you have is how it's going to present itself. But here's my rule of thumb. If you can pick it up with a stick, it's not it, right? If it clumps, if you can physically pick some of it up in your hand and it doesn't fall apart, it's not a HAB. So the four most common lookalikes from left to right is filamentous algae that grows, usually attached to rocks and in around slower moving water that have a lot of nutrients. Duckweed is actually, if you look really closely, it's small individual plants. Decaying organic matter usually gathers at the edges of lakes and reservoirs, but it's clumpy. You can pick that up with your hand or with a stick. And then early in the year, you're going to get a lot of lookalikes that are pollen. So, in spring, when leaf out is happening and all the pollen's flying around, that's going to be one of the most typical lookalikes. But if you look at it and it's got a orange or not orange, but like a yellow whitish tint to it, it's most likely pollen. So DEP, after that 2019 kind of disaster with Lake Hopatcong and the messaging and the economic impact of all of that, they really regrouped and came up with a pretty good system of messaging and making people aware of what's going on in any given area. So from left to right, watch, it has little infographics on there too. So you don't have to know exactly, what they mean. The graphic's going to tell you right away. So if there's a watch there, what they're saying is be cautious of swimming, be cautious of boating, but you can go ahead and fish. You can boat in a craft that's not paddle powered, so you're not kicking up spray. Once you move to advisory, then they start cutting back on things that you should be cautious of and tell you not to do it. Once you get to a warning, they pretty much advise you to not have a lot of contact with the water and so on. And then they do have this funny little one off to the right called Winter Watch. And as we've seen through this monitoring, there are some HABs that persist into winter. So there's a few genus that will persist into cooler temperatures and even in snow. So they can hang around. It's not just temperature, that is their determining factor. These are all available on NJDEP HABs website for they're downloadable. So if you want to be able to print any of these out to have any in your municipal buildings, they're all available for free. You can download the PDF there. So the best thing to do is, if you're going to recreate in an area, is know before you go, right? They came up with this really good interactive map, and it tells you when the last time it was sampled, what the reading was, if there's a watch there, if it's an advisory. And then again, they reiterate what the watch, advisory, and so on mean on the right-hand side. So they're going to say, okay, I want to go to Lake Hopatcong. I usually like to go fishing. Is that a good day for that today? You look that up and you go say, well, there's a watch, but that means I can still fish. If it was a warning, at that point, you're not going to want to go fish, and maybe you're going to look somewhere else, right? You look to go fishing maybe at White Lake or something different. So for our study, this was basically really volunteer driven. I asked the volunteers what they, were concerned about, where they were concerned about, and had them pick the areas that they thought that, they would go ahead and monitor the most. So these are the Lake Hopatcong sites. We were able to loan Lake Hopatcong one of our meters so that they could independently get a team together and monitor sites up there, along with a couple other meters from NJDEP. These are the Lake MUSCONETCONG sites. This is Saxton and TILCON lakes. And then also part of this grant was to look at agricultural ponds. So we met with NRCS and NJRC&D and created a list of agricultural producers in the area that might want to participate, whether or not there was any data on this previously, and what the feasibility of this would be. So out of all of the producers that I contacted, three farmers agreed to have their agricultural ponds tested on a weekly basis. And that's a lot, having somebody, show up on your property every week for 4 months, that's a lot. So I'm very gracious to these farmers for letting us onto their property. I'm not gonna identify any of them by name. We'll just go with, Ag Pond 1, 2, and 3. I told them I wouldn't share their data with anybody specifically to keep that proprietary. And so these 3 ponds were all a little different. And at first I was, like, well, how am I going to compare this data? But in the end, it kind of turned out to be a blessing. Ag Pond 1 was a producer that was aware of HABs as an issue, and she pre-treated her pond each season before temperatures started to heat up. She let all of her animals have open access to this water, so she knew how important that was to keep the water quality clean. The second pond that we looked at is a backup agricultural pond to water animals if need be, if the well system or anything like that, or for a drought took place. And then the third pond that we looked at was a pond that was used to irrigate crops, also in a drought condition. They had a bilge pump set up and used that as a backup water system in case of low water or drought. And then also some residents in Bloomsbury wanted to look behind the Bloomsbury Dam impoundment, so we took a look at that as well. So what did we find? Well, super proud. I was able to get over 50 volunteers interested and trained to do this. And I was able to retain 40% of the 50 people trained that actively monitored on a rotating basis over the summer. So I had promised I would retain 25%, and I was able to retain 40. And I attribute that to letting the volunteers kind of pick their spots, and that way there's way more buy-in. And it's going to be more local. They care about the water quality around them, right? So 4 months solid of volunteering, like I said, June through September, over 27 sites. Most of the time it was 27 sites, but some people also wanted to look at extra sites just because it was near their house, or they saw something that was of concern and wanted to monitor it. Over 555 samples taken to date. I still have one volunteer out there still monitoring on a weekly basis. Over 400 hours logged. And that just to me is staggering. That just shows you right there the dedication. And that includes the training. And then out of all of these 555 samples we took, there was 160 that exceeded the criteria that NJDEP lays out. So anything that meter read that came up 11 or over, 3 samples, right? We didn't just take one sample and say good enough. They would put it in the water and take one sample and then do it again 2 more times. And then whatever the average was between those 3 samples, if it was 11 or over, they would go ahead and report that to NJDEP on their report a HAB. It's like a Google form on their website, very easy to use. So here's the results. Here it is laid out graphically. So here's how it lays out in Lake Apacom. And if it's orange, it exceeded. And then I put the number of violations inside of that location, right? And up here, I think it's up here, no, up here, Cliff London is one of the, I have several all-star volunteers that are on this call today, but I wanted to point out, Cliff, I think this is up here is where you're monitoring on a pretty regular basis. But, a lot of these are people, like they live on the lake. These are near where people live. Like one of them, one of the sites is named Becky's house, or their dock number. But as you can see, this is pretty much every single site came up for violations, and not just 1 or 2 times over the course of the summer. And then I didn't even bother putting TILCON and Saxton Lake up here because there was no violations. We had no violations the entire summer in those spots. But if we come down to the agricultural ponds in Bloomsbury Dam, we see that we had 1 pond here with 13 violations over the summer and 1 pond with 6. And then I put this in green because this one was barely over the threshold. It was just like right at the 11 threshold on one of the warmest days of the year. And then Bloomsbury came up below the threshold every single time. So this was of concern. So this pond is near one of the tributaries to the Musconetcong. And, we were looking at this tributary for another project and came to, all of a sudden, my field specialist was like, it really looks like there's HABs in this water. It's pretty green. So we have the equipment and we went ahead and monitored it and, took a reading and it was off the charts, as well as, the readings up in the pond. So there's some kind of outfall pipe that leads from the pond to the tributary. And it stayed this way the entire summer. So this is, not an isolated incident of just an agricultural pond on somebody's land that's not harming anybody. There's a connection there. That water's getting into the tributary, and then the tributary is moving on down to the main stem, Musconetcong here. So we work quite frequently with Montclair State University, and we gave them a sample and they ran that sample for us, just as a favor because they were curious as well. And it exceeded the entire threshold of the test for toxin limits. It's not just having, you know, the HAB in there, which, you know, they're not all toxic, right? So you can have the algae bloom, but not all of them are toxic, but it came back extremely toxic. We were then able to alert that farmer to the situation because he was allowing, you know, his neighbors' cows to come over and flash graze in that area, and they had access to that water. So we wanted to make sure that, you know, no animals were harmed, you know, just being allowed to have access to that area. And that's an ongoing conversation at that location. So here how it plays out over time. So we saw how it looked spatially, right? So here's how it goes by month. So we started in June, and we had 9 violations, then 20, then 38, then 65. You can see the pattern, you know, what's going on there. Things are heating up, and there's way more sunlight available. And like I said, I do have an all-star volunteer that's out there still taking readings. And so far, from October on, there's been 20 more violations needed or to be reported. And so, you know, you know, we've been in conversation with Dr. Fred Lebnow. I think he's on with us today. And, you know, some of us assumed that once some of that warmer water had, you know, left the system, that we'd be seeing all of these HABs kind of decrease, but that's not the case. And, you know, I quoted him here, it's saying that there are a few genera that can tolerate the cooler, colder temps. Now, I'm not going to go ahead and say them. I haven't practiced saying those two. So, but you can read them there. And so this may be the case. You know, it would take some further study to figure out which ones are still coming up positive in Lake Hopatcong. But if Fred wants to speak after I'm done with the presentation, I would definitely welcome that. And as far as the supplemental samples that centenary students have taken to look at, there was a pretty high correlation with phosphorus, but we didn't take enough samples to really detect any type of trend. I mean, that is, you know, what the running analysis has been, is that higher levels of phosphorus, you know, that's basically their food, right? So you make it like your living room, you make it nice and warm, you give them lots of food, and they get nice and fat, right? Just like us. And so that's really no surprise. Another parameter that was looked at was pH. There doesn't really seem to be any correlation that we were able to find there, and not much of a correlation with the nitrogen. But like I said, it was a small subsample. And if we repeat this again, and the centenary students would like to use this as a student project over the summer, then, you know, I would suggest a few more and different sites to look at to get some more robust data sets. So on the MWA's website, we've created a web page so that you have access to some resources. It's under that tab, What We Do. And there's going to be links to everything that I talked about, that NJDEP map, you know, so you can know before you go. There's a training video there, so you can learn how to identify HABs so that you can then report a HAB if you see them. And then some helpful infographics down at the bottom that kind of, you know, just solidify all of this and like how HABs form and what you can do about it. So what can you do now that you've become a little bit informed? Well, you can get a little bit more trained up and learn how to identify HABs versus the lookalikes, and then report them to NJDEP. Personally, you know, you can use less fertilizer in your lawns and gardens, and even replace some of that lawn and garden with native plants that require less nutrients or no nutrients added to begin with. And they also have that added benefit if you live near a water body, that they leave a vegetated buffer, right? So that's going to filter out all of that runoff that comes across your lawn. And then become a river watcher and help monitor and become an advocate. So next steps, you know, I'm going to keep working with RC&D kind of to streamline that outreach process to farmers. for pond testing and treatment. There's been a few types of treatments discussed. Pre-treatment, like the one producer had used, seemed to be very effective, especially in smaller ponds, like most of these people have on their properties. Help other organizations expand their capacity with an equipment loan program, and roll that HAB training just into our regular trainings that we have in spring and fall before macro invertebrate collections, and just have it be part of what we do. This was all made possible through the Watershed Institute, so I need to give them their props. And if anybody has any questions, feel free to ask now, or you can, email me at musconetcong.org.
Thank you very much, Christa. That was excellent. Very interesting. I would take the opportunity now to ask if any of Christa's guests would like to make, she had alluded to maybe a doctor wanted to say something.
Hi, yes. My name is Fred Lubno, and really excellent presentation, Christa. Really good, and I really like to see this data. What's so nice about the phycocyanin is you're collecting real-time data, and volunteers can collect this very reliable information. I mean, it's the correlation is equating cell counts to the measurement of the phycocyanin. So DEP and we at Princeton Hydro developed those graphs. So that's where that 11 comes from. A couple things. One is, you mentioned those cooler water genera of cyanobacteria like Anthanozomenon and Celospherium. So while you were talking, I actually pulled up our phytoplankton data for HAPACON for this year. We're wrapping up our year-end report. The last time we sampled for phytoplankton was mid-September, but I'm looking at mid-September, and the dominant cyanobacteria were Anthanozomenon. So more than likely, that was the dominant genera that kept persisting into October and November. And just like in River Styx Crescent Cove, in mid-September, the cell count was almost 73,000 cells per mil. So it was up there in terms of those alert levels. And just two other things I do want to note: if you get some high phycocyanin readings, but it does not look like a cyanobacteria bloom, like if the water looks brown and you're getting high counts, I just want to, especially in the spring, I just want to give you a heads up. There is another group of algae called cryptomonads that also produce phycocyanin. We learned this with a bunch of lakes we're monitoring in Mercer County, that we were getting screamingly high phycocyanin, but we weren't seeing any cyanobacteria in the samples. And it turns out it's because of this small cryptomonad, which is actually a very good algae to have. It does not produce cyanotoxins, and it actually is a good source of food for the little animals in the water. So I wanted to throw that out. And the one question I had was, in that agricultural pond where you saw those really high counts, you mentioned measuring some cyanotoxins. Was it microcystin that they were measuring, or was
Yes.
it more than one?
No, it was microcystin. Yep.
Okay, yeah, and that's the most common cyanotoxin. So I was very interested in what type of cyanotoxin that they found. So again, thank you. Great presentation.
Thanks, Fred. Cliff, did you want to say something?
Yeah, if I may, great presentation, Christa. Some observations I made were essentially, and of the 160 or so samples that I took, close to 50% were over that threshold. But some of the sites, there was a large number of samples that were taken where we were over the number, but there was no real visible HAB at that point. You weren't seeing anything that would trigger a major alert. Some of the sites that started early ended early, and the sites that we were getting good readings through the summer become like right before Labor Day and through September. Once they started, they're the ones that continued. So I think it was a good exercise. I would love to see it continue next year. I'm still continuing to do it at the dock until we get some ice. But I think, I personally learned a lot about HABs, and it's great for the public and for the, people interested in the river to know.
That is a great point, Cliff. And I actually meant to say something about that, is that I don't have a number to put to it, but I would gather a guess that over 50% of the time that the reading on the the meter was at that threshold or over, people were not reporting seeing a visual HAB. It was there sometimes and it wasn't there other times. So you could be getting a high reading on that meter and not be seeing that visual component. So that is really important to know, that they can definitely be there without that visual, indicator.
And I actually had one instance where it was good on the meter, but I did have a visual green gobs in the water.
Yeah, and just to chime in, again, looking at the September data, I'm just seeing that we did have in River Styx Crescent Cove, we have an abundant amount of cryptomonads. So if you do see high phycocyanin, but it doesn't look like you have a lot of HABs, that might be part of the issue, that other group of algae.
Exactly.
Yeah,
Nancy? Oh, who is that? Sorry. Go ahead.
this is Rob, sorry. Yeah, I was just gonna add to that conversation. I mean, I definitely saw turbidity changes when the phycocyanin numbers skyrocketed. Definitely the clarity of the water just went down fast. So whether that was a HAB or not, I don't know, but I definitely saw a correlation between that. At least it seemed that way. I didn't do any analysis, but
You were using the Secudis turbidity tube, right, Rob?
right.
Okay. Nancy?
Yeah, I had a question. Are you and RC&D preparing any kind of an article or material to go out to producers?
That's
'Cause that
part
was
of the
very-
conversation. Yeah.
Yeah.
So we're internally discussing whether or not to take this a step further and apply for a SIG grant to create that process and create those outreach materials and look further into treatment methods, because we at MWA don't want to, have to go out and get a license to use chemicals. So that's a sticky wicket there. So we're researching the barley method because that is something that they could do. Also using those preemptive enzymes early in the spring in a pond that is a known troublesome pond for HABs is also a thought. So yeah, we're definitely having those conversations.
If Lebanon Township can do anything to, letter of support, please let us know.
Thank you. Keith?
Hey, yeah, great presentation, Krista. This is for you or for Fred, whoever might know. Just thinking about river users, is there any sense of how quickly either cell counts or cyanotoxin levels themselves actually kind of decay downstream of impoundments that have kind of HABs outbreaks? Like, is a river user 2 miles downstream potentially affected by blooms, or is it really at the spillway and not really too much downstream of that? Any sense of what that answer might be?
Well, I mean, just to chime in, anywhere, and Christa mentioned this, anywhere where the water slows down, I mean, that's habitat that the HABs prefer, but also that allows cells to accumulate. So like at Lake Apacon, you can be in the middle of the lake and things look fine in the middle of the lake. But as soon as you go to a near shore area, an isolated cove or bay where there's not a lot of movement, not a lot of flushing, not only is it better habitat for the HABs, but also the wind can concentrate the cells there. So in terms of a river user, like you're saying, if the water is flowing, it will have a less of an impact because there'll be fewer cells, there'll be fewer contact of the cells. Where it slows down is where you'll have more of an impact. Now, hydraulically, if you have a drought, a larger portion of that river can be impacted by the HABs just because of the hydrology and how, that flow rate goes down, and you may have some created slower areas. But like Christa mentioned, there's some run of the river systems that we work with that still deal with HABs, like large reservoirs that will have a flushing rate of only 2 weeks. But because of certain times of the year, and if they get in a drought situation, that can be exacerbated. So if you're recreating or dealing with flowing waters in general, it's better than if you're in a slower area when you're seeing those HAB occurrences.
So I see one more question from Nicole, and then we should probably move on to the rest of the agenda items. Christa, can you put your contact information in the chat in case
Sure.
people would like to follow up?
I guess I was, just listening to this conversation, and Krista knows I'm kind of into the whole native and looking at invasives, and I wonder how some of these invasives are having an effect on water quality. So I guess, my question is that as we're doing this, I'm just wondering if there's a way we can also look at the surrounding areas and like kind of look and see if there's like maybe like a lot of trees, the type of trees, if they're invasive, non-invasive, as well as some of the plants. I'm wondering if these things are also having an impact, as well as some of the microorganisms that are in the water. There are microorganisms that help keep these things more at bay. I guess those are some of my questions. Just things
That's a
to
good
think
question,
about.
Nicole. I'm not aware of any study that pairs together the presence, and the collage, you would say, of the plants that are present along with HABs. But I do know that when an area is more shaded and receives less sunlight, it is less prone to have a HABs issue just because they are photosynthetic. And so if you have an area that's become dominated, let's say by Phragmites or Japanese knotweed, and they've pushed out, the willows and the other taller trees that would be in the area, that would definitely be an issue because it's, out competing things that give more shade to an area. So I could definitely see that being an issue, but I don't know, Fred, if you know of any studies.
Yeah, the one thing I'll mention, unfortunately, it has not been identified in any, as far as I know, in any of the lakes in the Musconetcong watershed, but there have been a number of studies to show the very aggressive invasive species hydrilla. When it takes over, there are certain genera of cyanobacteria that are associated with hydrilla that can create cyanotoxins, and they've been documented to negatively impact waterfowl and other birds. So there have been a number of studies on that. So that's an invasive species. Number one, HABs aside, you do not want hydrilla at all. So I would definitely make sure that everyone knows how to identify hydrilla. It is just one of the nastiest aquatic plants, but there have been a number of studies to show that it's associated with certain types of HABs that can negatively impact waterfowl.
I was just going to add, I felt like in the past, I used to see more river birches along the rivers in the streams. I'm seeing less of them, and hemlock, I guess there's a disease out there that's been affecting them as well. So, I just wonder how these things are having an impact as well. And that's it. Thank you.